Okay, let's make a start. I know there's people still coming in, but since we're a little bit up against time, then let's get started. So it is a huge pleasure to introduce this year's Beddington medalist. So I think traditionally we say a little bit about Rosa Beddington, and then I'll introduce Guillermo. Could I get the slide of Rosa? So the first slide. Okay, so this is Rosa Beddington. This on this side is I can't see. On the left-hand side is a sort of formal portrait of her, which you'll be able to find if you do a Google search. And on the right-hand side is one that you definitely wouldn't be able to Google because it's of such low quality, but has a lot of the characteristics that I remember about Rosa, especially the cigarette in the hand and the air of interested discussion on whatever it was she was discussing at the time the picture was taken. The epithet kick-ass has been bandied around somewhat. For Rosa, that was entirely appropriate. When you read any of the obituaries of Rosa, because Rosa died in 2001 in her mid-40s, sadly, then words you read are things like scathing and didn't suffer fools gladly, so she was indeed scary and highly critical, but more than that, she was absolutely inspirational. So she was a passionate advocate of developmental biology in particular, and science in general. She was also a very willing listener to anybody who had a good idea, or anybody who had any idea. She would listen to you, she would give you her full attention, and she would give you full constructive criticism. And so I think it's a testament to the fact that she did that for so many people, that many of the alumni from her lab, or the people that she interacted with during her time in Edinburgh and Mill Hill, when I knew her and when I was a postdoc in her lab, have gone on to have productive careers in developmental biology. So she was a great mentor, and I think that it's entirely appropriate that the Beddington Medal will go to someone who works on gastrulation. His PhD thesis is right up Rosa's street. It studies the movements of cells during gastrulation, and mixes computational and experimental biology. And as Paul Martin said earlier, Rosa was an amazing experimentalist, and she was also a great artist. You would often see her down in the front row doodling, and one of her doodles appears on the Beddington Medal. So I think I'll ask Guillermo Serrano Najera to come up and receive his Beddington Medal for PhD thesis in this last year.
Thank you. Okay, thank you. Thank you for everybody. Thank you for this prize. And especially thank you for the invitation to come here. Actually, it was great. It has been a long time since I didn't have any of this kind of such an inspiring interactions with everybody. And well, today I'm going to talk about my main PhD project, which is titled the in vivo reconstruction of the main evolutionary transitions during vertebrate gastrulation. But before I would like to acknowledge everybody who was involved in this project. For the sake of time, is it working? Let me see. Yeah. So I want to just highlight 3 names. Of course, Case Bayer is my main supervisor. This couldn't happen without him. The second important name here is Manli Chuai. You will see a lot of data from her. She produced many of the experiments that complement my own results. And then Matthias Serra, who developed the computational models that you will see at the end of my talk. And of course, cannot forget the ISBio program and the University of Dundee. They really give me full support during my PhD and especially during the pandemic. So I did my PhD, as I said, in Dundee. And this city is not outside of this country. People don't know where is it at all. But actually it has a small, it has a page in the history of developmental biology because the book on growth and form by Darcy Thompson was actually written in Dundee. And I couldn't miss this because the first year of my PhD was the 100th anniversary of the publication of the book, so it was everywhere. And this is probably the most famous idea, although he explores so many other themes. So he tried to explain the variation in animal shapes using these grid transformations, right? So I think Darcy Thompson definitely would like the talk that I'm going to do now. And I'm going to begin with this. So recently I've been thinking a lot about vertebrate gastrulation. And as you know, gastrulation is this process when the animals internalize the mesoderm and the endoderm to create the inner cell layers. And this normally has like supermassive tissue flows and movements. And what is interesting is that very different organisms in different vertebrates internalize the tissue in different ways. And there are 2 very important differences between the vertebrates when you look at it in detail. So one of them, I think, is that the localization of the early mesoderm. So you have some organisms have these circles of mesoderm surrounding the embryo, while in amniotes, what we have is that the mesoderm has been restricted in the posterior part of the embryo. And the other key difference among all of these different organisms is that some of them internalize the mesoderm using individual cell ingressions, and that would be the case of, for example, chicken, but others internalize the tissue through involution, so basically using as a continuous epithelial layer. And that would be, for example, the case of Xenopus. So what I would like, so at the end of this talk, I hope that we will have a better understanding actually of how all of these different gastrulation modes or morphologies could possibly evolve during vertebrate gastrulation. But in reality, my project began with a slightly different question. I began trying to understand how cell behaviors are coordinated in space and time and over thousands of cells to generate shape in the animal embryo. And to do that, what we have is 2 important tools. We have a dedicated light chain microscope, and we have this transgenic chick line that expresses GFP only in the cell membrane, so we can evaluate the cell behaviors. And using these 2 tools, we can image the full embryo in vivo at cellular resolution during gastrulation. So before gastrulation, the chick embryo is just a flat disc of epithelial cells. And when gastrulation begins, the cells in the posterior periphery of the embryo begin to migrate towards the center and then along the midline, creating a groove, which is the primitive streak, and is going to mark the main body axis of the animal. Once the cells arrive to the primitive streak, they undergo epithelial to mesenchymal transition, they ingress, and then they migrate back to the periphery to create the inner cell layers, as you will see now after the movie. Yes, there. So let me explain a little bit how we think all of this is working. So we have the mesoderm, which is this crescent-shaped territory in the posterior region of the embryo. And this territory is characterized by 2 cell behaviors. So one of them is oriented cell intercalation. So in this case, what is happening in this territory, cells are interchanging neighbors all the time in the same direction, and this produces the convergent extension of the tissue. And the other is, as I already mentioned, cell ingressions. And if you combine these 2, you can have sustained a convergent extension of this territory, and as this territory extends, it pushes the cells in the rest of the embryo, generating these large tissue flows that we see in avian gastrulation. So how we think this is happening? So we observe that in these mesoderm territories, we have this, I hope you can see that, these long myosin cables that span several cells, and the contraction of these cables, we think, is what is driving the intercalation of the cells. And I just want to point this out now, but we will come back to this idea later in the talk. So the main idea here is that the shape and the cell behaviors of the mesoderm determine morphogenesis. And indeed, if you block the differentiation of the mesoderm, you completely erase any kind of morphogenetic flows, and basically it's a very boring experiment, and I'm not going to show it today. But what we will do later is I will show you how different chemical compounds can modify these two factors, the size and the cell behaviors in this territory. But before that, I would like to show you a little bit how we are going to analyze the data. And today we are going to focus in the use of velocity fields. We can have these very accurate representations of the tissue fluids, of the tissue movements on the surface of the embryo using a technique is known particle image velocimetry. And what is really interesting is that from the velocity fields, we can calculate a series of quantities which are the decomposed strain rates. And these quantities give us a lot of information of the underlying cell behaviors. The isotropic part of the strain rate, this is the technical name, is really the tissue growth rate. So this highlights those areas in the tissue which are undergoing expansion, and which areas in the embryo are undergoing contraction. And because this is a continuous epithelial layer, if it is undergoing expansion, it's because we have apical cell expansions and cell divisions, a combination of the two. And if we see a lot of contraction, it means that the cells are contracting apically and ingressing. And that's why the primitive streak is highlighted in blue. The other part of the strain rate is the anisotropic part. And really what is giving you is this shear deformation rate. And we represent it with these bars. These bars really what they highlight is they highlight those areas which are undergoing convergent extension. So basically the tissue is contracting along these bars and is expanding in the perpendicular direction without changing in volume. That would be the interpretation. And in these conditions, this continuous epithelial layer, really what this highlights is those regions where we have this oriented cell intercalation. And if we combine these two quantities, then we have a very good description of the cell behaviors that are underlying those tissue flows that we saw before. What I want to explain here is that what characterizes the primitive streak is that it has tissue contraction, cell ingression, and shear perpendicular to it. This means that the cells are intercalated in these regions. And these are the two main features of a primitive streak when we look at it with this technique. So using this, we can have these kind of movies where we can see how the embryo develops. Here we can see the deformation of this grid. And also we have the strain rates, sorry, which gives us, at any given moment in time, a little bit of the distribution of the cell behaviors and what are they doing in the embryo. And with this, now you can begin to do some interesting experiments. The first one I'm going to show was already done by Santa Saleb. He already spoke yesterday. So what they did is adding FGF to the embryo. And if you do that, what happens is instead of having a crescent mesoderm territory, suddenly you generate all this ring. And if you do that, you have the following situation. So these embryos begin to form normally. So you can see that the cells are moving towards posterior. So this looks very normal because this ring of mesoderm needs some time to differentiate. But as this ring begins to form, we can see that in these movies, the appearance of these circular structures surrounding the embryo proper. And this is when the mesoderm is mature. So if we look at this with our techniques, what we see is that this region is a ring with a lot of tissue contraction and shear perpendicular to it. So this means cell ingression and cell intercalation. Basically, this is a circular primitive streak. And we can see that using this transcription factor, SNAIL2, we are using it as a marker of the mesoderm. We can see that indeed all these cells differentiated into mesoderm. So, okay, this is a circular primitive streak, and then what happens when the cells ingress through it? So we explore this a little bit. So here the ingressed cells are highlighted in white. You can see that they ingress in this ring, and then they migrate towards the center of the embryo. And this is very interesting because this is very reminiscent to what is happening in the zebrafish embryo, right? So cells during zebrafish gastrulation, cells ingress in this germ ring territory, this circular structure, and then they migrate towards the animal pole that would be here on top of the image. So what we try to do is trying to reproduce the same results using a different set of compounds. And we decided to go from BMP inhibitions for two reasons. One of them is because BMP in the chick embryo blocks the differentiation of the mesoderm in the anterior part of the embryo. So this means that if you inhibit the inhibitor, you could expand the mesoderm territory. And the other is because these BMP inhibitors, in combination with this Wnt agonist, are used in vivo to generate mesoderm. So it could work also, in vitro. So it could also work in vivo. So we tried them, but we had some surprises. Now the things happen much faster than in the previous experiment. You can see large flows all around the embryo, and suddenly we have this large invagination that completely engulfs the central epiblast. If we look to these results, if you stop the experiment before this actually happens, you can see that indeed we produce a ring of mesoderm, so that was all right. But now there is a difference with the previous experiment, because if we look at this, we have a lot of shear in this region, a little bit disorganized maybe, but we don't have any kind of blue. We don't have apical contraction and cell ingression, right? So we look at it a little bit more in detail, and this is basically giving us the direction of the intercalating cells. And we are using, this measurement comes directly from tracking individual cells. And the IFGF case, the experiment that we saw before, the cells intercalate perpendicular to the ring. But in this case, the cells intercalate along the ring, tangentially to this ring. So what we think is happening here is that these cells begin to intercalate along this ring, and this, what it does is reducing the diameter of the ring, compressing the central epiblast. And when this tissue cannot be further compacted, the only solution that it has is to buckle inside and creating this large invagination that you saw. And of course, this looks very much like a blastopore of Xenopus embryo. In this case, I have a movie, so you can see that it looks very similar. But there is a very important difference. Like frogs embryos, what they do is internalize the ventral side of the embryo. However, here we are internalizing the animal side of the embryo, the opposite. Why is that? What we think is because the chick embryos have a massive yolk underneath. It's impossible that they internalize what they have underneath. And also all the edge cells of the embryo are attached to the vitelline membrane. And these two things impose this mechanical constraint, we think, make the system resolve in another direction. So instead of invaginating the vegetal side, invaginates the animal side. And this is the last experiment I would like to show you. This is VEGF inhibition. So we noticed for other reasons that blocking VEGF signaling produced these fat, short primitive streaks. So we decided to mount this in the microscope. And we see the following. The beginning, everything looks more or less normal. Cells are flowing towards the midline. But then after sometimes the tissue flows begin to slow down, and instead of creating this very well-defined primitive streak, it creates something like a triangular structure, like this. I don't know if you can see it very well. I have a trained eye. And then they end up creating this a small invagination in the front of the forming primitive streak, if you want to call it like that. So if we look to what is happening, what we observe is a lot of shear. This means that these cells are actually intercalating and going to the midline. However, we lack the blue signal characteristic of the primitive streak. So in this case, cells cannot ingress. And in these conditions, what we think is happening is that more and more tissue is arriving to the center, but we don't have a sink. So the tissue begins to accumulate, and when the epiblast cannot accommodate any more cells, then the only thing that it can do is actually to buckle and create this little invagination at the front. This kind of resembles what we see in gastrulation in chameleon by Stower, probably here as well. It's 2015. We were discussing this. These chameleons gastrulate in a very interesting way because they internalize part of the tissue in this structural blastoporal plate through individual cell ingression, but they also internalize part of the tissue through involution, and in this structure known as the blastoporal canal. So it's quite interesting that if we reduce the amount of ingression in the chick embryo, suddenly you can create this little invagination as well in the front of the primitive streak. So first conclusion, we generated this kind of thing. So we can generate a crescent with ingression. This is the normal case, primitive streak. You can have a crescent without ingression, and you generate this little invagination. You can have a ring of mesoderm with ingression, and then you generate this circular primitive streak. And you can also generate a ring of mesoderm without ingression, and then you generate this blastopore-like invagination. So it looks like there are two parameters controlling these morphologies, the amount of mesoderm and if the cells can actually undergo ingression or not. And we tried to test this idea using modeling. So we contacted Matthias Serra in the Mahadevan lab, and they created this model where the embryo is represented as a viscous compressible fluid, and myosin, those cables that we saw in the beginning, can generate, that are associated with the mesoderm territory, can generate stresses that move the tissue. And the interesting part of the model is that the myosin itself, we know this for other experiments, is itself mechanosensitive, and that is also included in the model. So tension can induce the propagation of these myosin cables. It's a continuous model of partial differential equations. We can come back to that. So what we did is actually, Malia and me, we quantified the direction, the different patterns of myosin with these different treatments that I showed to you. But at the end was very simple. In reality, it's like there is only two things. You could begin with a crescent, and you can begin with a ring. So it's exactly the same as the mesoderm territory. And then in this model, basically, what they did is begin in these two conditions, and then just they modified one parameter, P0. And P0 is really the ratio between the isotropic and anisotropic contribution of the myosin to the tissue movements. But what we can do for the biologists, this is really a kind of a representation of the cell ingression. So if you activate, basically, if we, with only these two conditions, changing the initial condition and changing if the cells can undergo ingression or not, they can produce all these different morphologies. As you can see, this is the model, and these are the measurements in my experiments, and they have an excellent agreement. So very interesting. So this is the last slide. What we see is that, yes, it looks like by basically moving, changing these two parameters, the amount of mesoderm from the crescent to the ring, and changing as well the amount of cell ingression that cells in the mesoderm can undergo, then you can visit in the chick embryo all these different gastrulation morphologies. So this, the conclusion for me is that this is very interesting because by doing relatively small changes in signaling, this can propagate through their effect in cell behaviors into producing large morphogenetic changes, and this might explain how all of these morphologies were produced during vertebrate gastrulation. So just before I finished, this award was for my PhD, and of course, it would be absolutely impossible without one person. Of course, this is my supervisor, Kees Beyer. And I just want to thank him. He has been an incredibly supportive and inspiring supervisor. And I just want to remember one anecdote. In my second year of PhD, I actually asked him, what is the best piece of advice that you could give to a PhD student? I think it was a good piece of advice. Apparently, he did a good PhD. And so I want to share it with all the PhD students in the audience. So he told me, you have to be pragmatic and optimistic, love what you are doing, and sleep well. Thank you.
Thank you very much. That was great. Do we have some questions from either the audience or online? Can I start? Or unless there's there's one. Jeremy.
Really beautiful stuff. I loved every second of that. Thank you. So one of the components that doesn't seem to be in the models, but of course is in real life, is involution.
Yeah.
And you sort of created an involution with your one of your ring situations. So I mean, I was just trying to reflect how separate a sort of module. What's the difference between involution and ingression? Which sounds like a trivial question, but in a way I want to ask it the other way around, which is, have you thought about how similar they are? I mean, have you thought about how the sort of aligned ingression really looks like involution at a sort of cell behavior level?
Yeah. I'm going to answer this question with more biological perspective, if you want, because I mean the modeling. So what we can see, one of the difference, I think, is that when we lose cell ingression, what happens is that the tissue in the epiblast cannot remain under tension, and that's why it buckles. So every case, the BMP inhibition case, the VEGF signal inhibition case, what happens is we don't have ingression, and in this case is when we have folding. So I don't know if this is different at the mechanical level. So so I don't know. I don't know if this answers your question, but
I think it's something to think about. I mean, again, I'm just sort of throwing it out there.
Okay. Any furthers? Yeah.
Thank you. It was a really good talk, and it's somewhat similar actually, because so in a frog you have involution all the way around the dorsal lip. I think it was Ray Keller and David Shook showed that in, say, an axolotl, and I believe in some other basal vertebrates, you get both. So you get maybe on the ventral side ingression and on the dorsal side involution.
Yeah.
So where does that sort of fit in your model? Why would the cell choose, say, maybe to ingress at one point and then maybe involute in the other?
I think it could be possible. Maybe there are more morphologies here. Maybe this is not a complete morphospace, right? I think maybe it's possible to produce different kinds of mesoderm, so and maybe in one part of the embryo they could actually undergo ingression, and in the other part of the embryo couldn't. But basically, the way we are adding these inhibitors is just everywhere. So maybe we cannot generate that kind of structure.
It was really impressive data, though. So thank you.
Thank you.
Can I just jump in and ask you something?
Yeah.
I wonder whether you think that you have mechanical forces that are shaping the shape of gastrulation. Could you make a frog-like chick by putting a chick epiblast on top of some kind of yolk-like substitute?
I don't know if it would be possible. I mean, it would be very interesting
to
see what part is involuting. I don't know.
Do you think that that just sitting a chick epiblast on top of something a little
sphere
smaller,
or something, it would be very interesting to try to modify the. It's actually a very interesting idea. Just to try to force this flat embryo into a sphere or something like that and see what happens. I don't know.
Oh. There's one more? Yeah.
Yeah, it was a fantastic talk. I just wanted to ask, maybe this is naive, but so you've sort of explained how you can get similar mesoderm internalization in different ways. I was wondering, do you know what the differences in the outcome, or what's the selective pressure might be to actually give you the mesoderm organization?
The main theory here is that is the increased size of the yolk. So once the embryos began to accumulate yolk, for example, to lay egg on land, this massive yolk imposed this constraint into embryos, and they became flat. And this is what probably kicked the. Well, although, I don't know. So but also the self-fish embryo. But yes, in general, we think is because of something to do with the geometry of the embryo and adapting to the yolk.
with the chameleon or something, is there other, for example, other sort of selective things about when the mesotome goes in, how that's patterned if you do circular or involution or ingression?
Yeah, probably. I don't know.
Thanks anyway.
Okay, well, thank you. Let's just thank Guillermo again.